Hello, and thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's scientific briefing is one in a series designed to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Our expert speakers, Doctors Silvia Crescioli, Shashi Jatiani, and Jason Lohmueller will be discussing precision antibody engineering for solid tumor immunotherapy with us today. Please note that this briefing is being recorded. You can add any and all questions to the Q&A box or to the chat in the viewer, and the questions will be answered at the end of the presentation. And feel free to address the questions to the speaker specifically if you would like. And without further ado, I will turn the show over to our speakers.
Hello, good morning or good afternoon, everybody. My name is Silvia Crescioli, and I'm here today to give you a brief introduction to Dr. Shashi Jatiani and Dr. Jason Lohmueller presentations. As you know, the field of antibody therapeutics is rapidly growing. We currently have 214 approved antibodies globally. 36 are in regulatory review in at least one country or region. 209 are in late stage of clinical development, and around 1,100 are in early stage of clinical development. And as you know, this is just the tip of the iceberg because there are many more antibody therapeutics in preclinical development that unfortunately I cannot keep track of. So if you look at the antibodies in late stage studies and you stratify them by therapeutic area, you can nicely see in this pie chart that all the major therapeutic areas are represented. Of course, you have half of the antibodies for cancer indications, a quarter for immune-mediated and inflammatory disorders, then you have a 6% for cardiovascular and hemostasis disorders, and then you have antibodies for infectious diseases, ophthalmic, metabolic, neurological, skeletal, and muscular disorders. And this is to show you that how antibody therapeutics have now become a very versatile platform that can be potentially used in any disease area. And why is that? This is because antibody therapeutics can be designed with many different mechanisms of action. They can be designed for binding a receptor and block the receptor or activate the receptor, or they can bind the soluble factor and neutralize it or promote its clearance. Or because of their Fc portion, they can engage the immune system and trigger Fc-mediated effector functions. Or still via the Fc portion, they can be internalized by antigen-presenting cells together with the target antigen and facilitate antigen presentation. Or they can be designed for cell engagement, and this is the case of certain types of bispecific and multispecific antibodies. And last but not least, they can be conjugated to payloads, which could be small molecules or tags, and this takes advantage of the targeting property of the antibody, so to deliver specifically to the diseased area a certain payload or a tag. And this is usually used to improve the therapeutic index of a payload or of a therapeutic modality. And you will hear more about this later during this webinar. So antibody therapeutics can act via one or multiple of this mechanism of action. And nowadays, antibody engineers that can be design their antibody with intentionally with certain types of mechanism of action. And they can also design their antibody not to have other types of mechanism of action that may impact efficacy or safety. And how do they do that? They do that designing the Fab regions of the antibody and the Fc regions of the antibody. And here are listed all the major considerations that are done during Fab or Fc region design, but I can't go through every detail. What I'd like to point your attention to is that, of course, Fab design will influence the antigen binding properties of the antibody and therefore the direct effect. And the Fc design will influence the affinity of the antibody for Fc receptor and the C1q component. And this translates into the capacity of the antibody to trigger Fc-mediated effector functions, but also the safety and the biodistribution of the antibody. And then there's another concept that although Fab and Fc regions exert two different functions, they are closely connected. And nowadays it's widely accepted that one can influence the function of the other and vice versa. So this is quite important because Fc design, especially, has been historically overlooked, especially for certain antibody therapeutics modalities, while now is widely accepted that is a central core part of antibody therapeutics design. And to give you some context in terms of the Fc design in the clinical pipeline, here I'm showing you the isotype choice in approved and late stage studies antibodies. First of all, you can see that the majority, 95%, do have an Fc. So only 5% don't have an Fc. Also, 60% have a human IgG1 Fc, followed by human IgG4 Fc. And then if you focus on the antibodies with an Fc, and you look at the Fc engineering status, you can see that nearly half of these, at least, are Fc engineered. And they can be Fc engineered for many different reasons. Here I group them all together. They can be for Fc silencing or for enhancing effector function, for half-life modulation, or in the case of IgG4, for hinge stabilization, or for heterodimerization. And this is the case of certain types of bispecific and multispecific antibodies with an asymmetric format. Or for antibody conjugates, they can be and Fc engineered for site-specific conjugation. So here I'm going to give you a brief overview of the main strategies for modulating effector function and half-life in terms of Fc design. If you don't want your antibody to exert effector function or you want to reduce them, you can choose an isotype like IgG2 or IgG4, which have minimal interaction with activating Fc gamma receptors and the C1q component, and therefore they have poor capacity to mediate effector functions like ADCC, ADCP, or CDC. Or since they, especially IgG4, they do have a little bit of interaction with Fc gamma receptors. So in certain contexts, they are able to trigger effector function, or at least their binding to Fc gamma receptors can impact safety. There are other options. You can perform protein engineering, and for example, you can generate hybrid antibodies with Fc gamma receptor binding domains of IgG2 and IgG4, or you can start with an IgG1 backbone and introduce point mutations to reduce or remove effector function, and this is the most used strategy. Or you can start with an IgG4 backbone and introduce point mutations to remove the minimal residual effector function that IgG4 have. Otherwise, another option is to perform glycoengineering. So you can do that via protein engineering. So introduce just one single point mutation and remove the N-glycosylation site, or you can chop off the glycan. And in both cases, the result is an aglycosylated antibody, which has been shown to have minimal binding capacity to Fc gamma receptors and therefore poor capacity to mediate effector functions. Instead, if you want your antibody to have an effector function, first of all, you can choose an IgG1 isotype, which is able to mediate effector functions like ADCC, ADCP, or CDC. Or if this is not enough, you can perform protein engineering and introduce point mutations to enhance the binding to activating receptors or reduce the binding to the inhibitory receptor. Or you can perform glycan engineering, for example, the fucose removal. Afucosylated antibodies have been shown to have increased binding to Fc gamma receptor 3A, for example, and so be potent mediators or of ADCC, but also other types of effector function in certain conditions. Or you can design your antibody for modulating the half-life. So now most of the antibodies are designed for when most of the half-life modulation strategies are for half-life extension, although there are some approved or late-stage studies antibodies with reduced half-life. So for half-life extension, you can perform protein engineering, and the idea is to introduce point mutations to improve the affinity to FcRn at low pH. So now, this is my concluding slide. With this one, I just want to show you, now that I showed you how you can design your Fc, all the different strategies to improve the effector function of the half-life, I want to show you the many different types of cells that express Fc gamma receptors and other types of Fc receptors, and that can therefore interact with your antibody. So the balance of activating and inhibitory receptor and the level of expression of these and the affinity of your antibody for these different types of receptor will affect the capacity of your antibody to trigger effector function and which type of effector function to trigger. So the system is quite complex. So in summary, I showed you how antibody therapeutics have evolved into a very versatile platform that can be potentially used in any disease area. Showed you the importance of Fc design and engineering that is now central for optimizing efficacy and safety. I showed you how Fc-mediated effector function are complex, the different players, the variety of immune cells expressing a variety of Fc gamma receptors. And so given these, the functional behavior cannot be fully predicted by the design alone. And therefore, a broad functional screening appears to be essential for a proper characterization of your antibody. And now I'll hand you over to my colleague, Shashi, who will tell you more about it.
Thank you, Silvia, for a wonderful overview. I think we've been, well positioned with this overview to see how isotypes matter, engineering matters. Conjugates are harder to predict than naked antibodies, but even they are not fully predictable. And then there's complexity in how receptors and which cells express them, how they express in which. And one more point is that sometimes these are inducible expressions, so context matters, disease biology matters, physiology matters. So what I want to talk about is some deep profiling. When we say deep, it's exactly where Silvia left off, is broad Fc profiling, not stick to just the three famous canonical Fc effector functions in ADCC, ADCP, and CDC. And the use case that I'm going to show you of our platform at SeromYx is for approved HER2 targeting ADCs, and then I'll set up the stage for Jason, who's the featured speaker for today. So this is something that we love to look at and show to our friends in the field, is that we now have a very amazing diversity of antibody formats emerging, and some of them are very, very hybrid. I mean, you take all these elements and you can make them even further partnered with each other, and everything in the blue rectangles has Fc in it. The antibody fragments don't, but everything in a blue rectangle in here, in these three rectangles, have an Fc. And when you look at what that diversity looks like, it's quite impressive. You have Fc engineered, you have conjugates, you have isotypes, not just IgGs, but now IgEs in the clinic, IgMs in the clinic. There are attempts to make hybrids called IgEs, etc. You have polyspecifics, bispecific, multispecific, Fc fusions, hybrids, right? So it becomes very complex, very fast. And it's not because people are having a field day in trying various things in their sandboxes, because each of these have an advantage over another format. And sometimes you just have to try many things to target a difficult target or try and treat a difficult disease. And so amongst the conjugates, you have ADCs, which are the quintessential conjugates. You also have oligonucleotide conjugates, AOCs. You have ISETs, which I mean stimulatory. And then you have other conjugates that are coming up, including bifunctional with small molecules. And, it just keeps getting more and more diverse and unpredictable. And increase in complexity increases the need for comprehensive empirical testing is what we believe at this stage. We know we are heading towards a world where you could take a sequence, put it on a large language model such as FCGPT, which is an excellent start, but not there yet. And you'll have enough validation from experimental efforts to have models that can predict FC function as soon as you enter a sequence. But we're not there today. We will get there one day. And so today is the day of empirical testing. Okay, so yeah. At SeromYx, we have what we think is the broadest Fc effector function profiling platform in the industry. And in pink rectangles up top, we have the biophysical assays where we look at the whole Fc receptor binding array. Now, why is this different from what others do? Is because this is antigen specific. It's a 3-party, not a 2-party. It's not just the Fc of an Fc receptor and the Fc of the antibody being assessed. It's being done in an antigen specific way. We have antibody isotyping subclassing and avidity. That's more for use cases where you use polyclonal samples, such as patient serum samples. And then, of course, we can do antibody Fc receptor affinity, both 2 parties, just the receptor and the antibody, or we do have now developed a system by which we can look at 3-party. That's been a heavy pull, and we are excited to show that data in the upcoming publication. In blue are all our functional assays. Some of them are acellular, such as ABCD, which is another way of looking at complement deposition and therefore leading to activation. But you have the 3 canonicals in there, ADCC, CDC, and ADCP. But we look at other cell types, such as eosinophils, dendritic cells, basophils. And some of our assays are more infectious disease specific, such as mucin binding and enhancement of infection. But this is a broad array. It's by no means an entire set of Fc receptor Fc interactions that exist. I think the grid can go up to 51 different functions, but we have robust industrialized standard assays now with very good metrics on robustness and on an industrialized platform. So a developer can come and either choose the whole set or pick and choose from this menu of assays that are available. We focus on physiological relevance. I already mentioned antigen specificity. That means tripartite, because that's what one would expect in vivo. We use as effector cells from healthy human volunteers. We only use human cells. We have a high throughput platform. We can do thousands of examples in a single experimental run. And it's a very adaptable platform. We've tested it against many antigens, different formats of antibodies, matrices, including nasal swabs, and of course serum samples, purified antibodies, different subtypes and species also can be tested on our platform. When you generate this amount of data, sometimes it's just good to send this through a very strong computational pipeline, and we have that. We can look at all this data and send it through a platform that's well established and help developers with decision making. Ours is the only CRO that I know in the world that has only FC focus, and therefore we have deep expertise, and we are a GCLB accredited facility. Just a quick show of what our platform has been used for very recently, since 2005 to today. Here are some publications. This review was written with Silvia, our first speaker, as first author, and Lenny Moise, our CSO. And we wrote about how broad profiling is a powerful way to de-risk antibody development, and it's been well received and even quoted in international conferences. Here's an example of a use of our platform, IgE antibodies. This one against, a specific target called folate receptor alpha was tested, and we came up with deep insights on this mechanism of action on our platform in a collaboration with KCR. In another collaboration with Sophia's lab, we went ahead and showed how Fc-engineered glycomodified antibodies can be tested, and you can parse apart the activities that these are engineered for between the Fc variants. So our platform is sensitive and robust in that sense. And just today, we were accepted in Cell with a powerful collaboration with Kiko Iwasaki in Yale for this long COVID study. This is more a system serology serum sample-based study. We're so excited that's happened. The publication date is yet to be. I think it's towards the end of this month, but or the next month in May. But you have fresh news that we have just been accepted in Cell. Today's topic, we're answering internal data, and this is not in collaboration. This is just taking approved HER2 antibody trastuzumab and its two approved antibody drug conjugates, TDM1 and TDXD, and comparing them side by side on our platform. And so today I'll be presenting mostly biophysical data and one functional, and this is an ongoing, so this is hot off the press, but of course, phase 2 of the study is all about the functional assays that we do and what outcomes we see as we compare these very well-established marketed drugs. So, just a quick show, of course, trastuzumab is not a conjugate, it has a drug antibody ratio of 0. TDM1 3.5, and TDXD has a DAR of 8. Once considered a first gen, second gen, third gen HER2 targeting antibody format, right? So we have a specific set of controls. For trastuzumab, we have IgG1. For TDM1, we have IgG1 with TDM, the exact drug antibody ratio, and similarly for TDXD. As a negative control, we have a recombinant, as an antigen, we have recombinant human HER2. The references for what's on this table are at the bottom right. So when we look at Fc receptor binding array, we look at Fc receptor 2A, both alleles, and you see what is expected. You see robust binding of trastuzumab and its ADCs. There's slight rank ordering here, which tells you that trastuzumab is the strongest, and that's expected because there's no interference from the drug conjugate. And the controls look really good for all three maps. They are flatlined. It's not that we didn't do those experiments. Those actually have 0 values. Similarly, we looked at 2B. 2B is the only inhibitory receptor, and all 3 antibodies bind roughly equally, and the controls are negative. We looked at 3A, both alleles. 3A is important. It's on NK cells, and it's for ADCC activity. Trastuzumab does well here. TDM1 does quite well, and TDXD is not so great. And this matches what's expected from literature surveys, what is known about these antibodies. And both alleles have been tested. The 2A receptors, of course, are for ADCP or cellular phagocytosis. ADNP, that's neutrophil phagocytosis, has contributions from 2A receptor binding, but also from 3B. That's the one that's unique to neutrophils. And here's a prediction from our biophysical analysis that trastuzumab may have some ADNP activity. TDM1 is expected, therefore, to have a little less, but TDXD is probably going to have minimal or a lot less ADNP activity. Again, the controls look excellent on this data set. We looked at FcRn binding, and this is known. There's a rank order between these 3. Trastuzumab is supposed to have the longest half-life. TDM1 follows, and TDXD follows that. And you see that rank order maintained at pH 6. And there's no effect on recycling at physiological pH 7.4, and that's expected. And then this is just Fc receptor 1A binding, which is not a distinguisher of how much activity you'll have, but it's like it's a prerequisite for Fc effector function, and all 3 do bind. So Fc effector function is expected from these various versions. Here's an interesting one that threw us off on a bit of a head spin. We looked at C1q binding, and trastuzumab bound to some extent, and TDM1 super exceeded that. T-DXd was lower than trastuzumab, and it made us really worry. What does this mean? Because in literature, trastuzumab does not induce much complement activation, and TDM1 has not been known to be associated with C1q binding, with the complement CDC activity. So I did a literature survey, and this is what we found, that there are differences amongst these. And what I want us to focus on is on the lower half of that table, the biophysical characteristics of these antibodies change based on what is conjugated to them. There's low hydrophobicity on trastuzumab, high on TDM1 because there are DM1 patches that come up, and very high, in TDXD, which changes a few things. You have, let's look at just the last row. You have clean docking, so to speak, of C1q in trastuzumab's case. TDM1, potentially sticky interaction because of this high DM1 patch situation going on. And then there's physical shielding of the CH2 domain with TDXD. So the chemistries are different, and therefore the biophysical characteristics are different in this biophysical assay. So what is the tiebreaker? We went ahead and did the ABCD assay, where we're looking at the deposition of one component of the complement system. And here we go, trastuzumab and TDM1 look a lot like each other. TDXD looks a lot lower binding. And what would be predicted from here is what will match literature. What is known in literature is trastuzumab may have some, if any, activity. TDM1 cannot exceed trastuzumab, and TDXD will have zero binding, or CDC activation. So that's the data set. It's exciting that our platform has broad FC profile, application even in ADCs, and we've seen a baseline for how different payloads and linkers alter the FC profile from this data set. We also validated the fact that FcRn binding profiles agree with the known half-life of these in rank order at least, and then the drug antibody ratio and surface charge govern how effectively an antibody or its ADCs recruit C1q, but that is not always reflective of ABCD or C3 component activity. And then I just pointed that out, that we, from our biophysical analysis, expect CDC activity to be, if any, highest in trastuzumab. I want to set the stage up for our next speaker, Jason Lohmueller. We have this very exciting collaboration going on with him. He has a fantastic lab in UPIT. And what his group has done is they've tried to make FC-modified antibodies, which will direct CAR T cells to the site of action. So the antibody is either trastuzumab or pertuzumab, and it targets HER2, and then they have this linker through which they add a synthetic motif. How does that play out in biology, in cell culture and in vivo? Well, you have an immune effector cell expressing Fc receptors, the Fc receptor shown in dark blue. You have the cancer target cell expressing HER2 in red, and you have a CAR T cell which has a CAR receptor that's specific for that synthetic motif, and is the antibody that's central to this interaction, bringing all three types of cells together. What we are looking at with Jason was which is the best linker in terms of the length, the flexibility, and the site of conjugation. And we ran a set of assays on our platform, and I will let Jason tell you about it. The idea really is to de-risk CAR T cell fratricide and unnecessary immune activation, but also to see are there synergistic activities in addition to CAR T cell. So I'm going to stop sharing, hand it over to Jason.
Excellent. Thanks so much, Shashi, for the great intro. And let me get my slides up. All right. So I'm very excited to tell you about some of our work today on universal CAR development, and especially our new adapter development strategies, which are really given a boost from our collaboration with SeromYx and their great technology. So first, these are my disclosures. And I'm just going to take a brief time to dive into background on CAR T cells, or chimeric antigen receptor T cells. So these are engineered T cells that express a chimeric receptor, and this binds to an antigen on a target cell, most often a tumor cell, and then leads to signaling in the T cell, and then lysis of target cells, T cell proliferation, and different effector functions like cytokine production. And this is administered as an adoptive cell therapy. So the cells are manufactured, most often isolated from the patient, then engineered with a viral vector to express the CAR, expanded, introduced back into the patient where they can hopefully find and kill the tumor cells in the patient. And this has been a very effective therapy for hematological cancers. There are now 7 FDA-approved products, 5 of them targeting CD19 on different B-cell leukemias, lymphomas. And excitingly, these have really been curative for a large number of patients with B-cell lymphomas or CLL, and also often curative for ALL as well. And the second group of therapies are targeting multiple myeloma, BCMA directed, and these have also been highly effective. And it's a little bit early to tell about long-term durability, but this also appears very promising. And here you can see just the different CAR constructs in those patients, in those products. And really the focus of my lab and the technologies that we're developing is trying to get after the limitations of CAR therapy. So these stem from manufacturing failures, so the inability to produce enough cells or the cells not persisting in patients. Another type of failure is antigen modulation. So for any targeted therapy, if your disease loses the antigen, then you could have disease relapse and resistance. And then there are also issues of toxicity. So this is a highly potent therapy, just a massive immune reaction acting in the patient's body, leading to cytokine release syndrome or other toxicities like neurotoxicity. And then finally, there's also a huge unmet need for other types of diseases, different lymphoma subsets, but also solid tumors, which make up the majority of the cancer epidemic. So this is really where, again, my lab is focused, and one of the main technologies we're developing are universal CARs. So what these are CARs that instead of directly binding to a target antigen, interact with one or more adapter molecules. So in this case, antibody adapters that have some antigen binding, and then also have some CAR binding activity, and in this case through a tag. And so upon interacting with the adapters and the antigens, this leads to the same effector functions from the CAR T cells, activating them, lysing the targets, and other effector functions. And overall, this has several key advantages over traditional CAR therapy. So you can get tunable activity. Instead of needing to kind of titrate the dose of the cells, you could actually give a high dose of the CAR T cells, but tune the activity by the dose of the adapters, which gives it a nice extra point of control. You can also target multiple antigens on the same tumor cells using the same CAR T cell product, simply by adding multiple different antibody adapters to the patient. Similarly, you can also use the same CAR T cells to target different types of cancer in different patients, again, by combining them with different antibody adapters. So that's where this universal term comes in for the technology. And just briefly, the way this is administered, so very similar to the T cell production, it's very similar to standard CAR products. Through, in at least current forms, through leukapheresis, viral transduction, then infusion back into the patient. At the same time, there's this extra point of control where the adapters are infused into the patient to, again, control and direct the CAR T activity. And we weren't the first to come up with this type of system. There are many in the literature, and I think that shows the excitement around the technology. And the way they really differ is in how the CAR recognizes the anti-tumor antibody, and what is the tag, what is the CAR binding protein. And so these have ranged from avidin biotin, different protein-protein interactions, including leucine zippers or binding to peptide neoepitopes. So there's really a broad array of these technologies in development. And I do want to highlight, so even though it's still somewhat early days, a number of these have actually made it to the clinic and have shown some early promising clinical results. So this is some data from Caliber, where they compared an anti-CD19 universal CAR with some of the existing trials with the FDA-approved products here. And they saw pretty similar overall response rates, but they excitingly saw a faster time to resolution for some of the toxicities, so both cytokine release syndrome and then some of the neurotox compared to those previous therapies. So I think this therapy has some promise for cancer therapy. And so where we kind of entered the fray here is that we developed a new technology using this SNAP tag protein, which recognizes a benzylguanine tag. So this is a human enzyme that's been modified so that it will specifically interact with benzylguanine instead of the native 0,6-methylguanine. And the really exciting thing about this is that instead of simply binding to the antibody adapter, the CAR actually forms a covalent bond with the adapter. So we're actually assembling the CAR on the surface of the cell. And so when we started developing this, we had a design that uses it's a second generation CAR design using the 4-1BB signaling domain. We added in the LNGFR expression marker, and we're using a retroviral construct here. And we showed we could get very high expression of the CAR on human primary T cells. And on the adapter side, so we took kind of usual suspect antibodies that have been in the literature, have been in the clinic for a long time, including rituximab targeting CD20, FMC63, which is the antibody used for the all FDA-approved CD19 CARs, cetuximab targeting EGFR, Herceptin targeting HER2, and then we also, in addition to the full-length IgG antibodies, made a Fab fragment of the rituximab. And to make the conjugates, so we ended up using random lysine conjugation using an NHS ester. And this is similar to some of the approaches used for adding, for instance, drug payloads to antibody drug conjugates. It's one of the modification or conjugation methods employed. And so we then quantified the number of BGs per antibody, and we had about 2 to 3 using this method. We could dial this up or down using different amounts of the reagent, but in general, this is where we landed for our first set. And then one of the early assays that we wanted to do was to see, could we actually retarget cells with the SNAP CAR to different antigens by incubating them with the different antibody adapters that I just went over, as well as different antigen targeting or antigen positive cell lines. And so you can see if we incubated the SNAP CAR cells with the antibodies, but with no target cells, we saw no activation. So just having the adapter there isn't enough to activate interferon gamma production from the T cells. But if we mixed the antigen positive cells with the different adapters, only when we had antigen targeted adapter plus the antigen positive cells did we see induction. And we saw very robust activation, including with the full length as well as the Fab. And we saw this for also with the EGFR and the Herceptin here, and HER2 targeting here. Next, we also looked at specific lysis of the tumor cells. And again, we saw very robust antigen-directed activity from those adapters. And we've now gone on to, I think we've tested now 25, or actually a couple more different antigens and antibody adapters. So we've found this to be highly programmable across many different tumor antigens. Another thing we wanted to see was, could we actually kill cells based on multiple antigens at once. So we mixed in, these are single tumor controls. So we had the Raji cells, which are CD20 positive, which we could efficiently target with rituximab. These are K562 cells engineered with EGFR, and we could efficiently lyse them with cetuximab. And then we did combinations of the cells, and we want to see, could we actually specifically kill those different cells with the adapters? And so when we had the mixes, but when we looked by flow at the individual cell populations, if we had no antibody, we didn't have much lysis. If we had rituximab, which targets CD20, again, these cells weren't significantly lysed, but when we had cetuximab or the combination, we saw high amounts of lysis of the cells. And similarly, when we, in the same mixes, if we looked at the Raji cells, rituximab and the combination were very efficient at killing those cells. So this is great that we can kill tumor cells in vitro, but we are interested in applying this to human tumor xenografts in mice. Actually, before that, we want to see, could we actually, with the SNAP tag, the SNAP CAR function, would it actually be labeled by adapters in mice? So we took the rituximab benzoguanine adapter, injected it IP, and we injected the CAR T cells in retroorbitally, so through a different route. And then we, one day later, went to see would the T cells be labeled by the adapters. And excitingly, what we saw, this is looking by flow at LNGFR, so CAR expression on the X, and then antibody labeling on the Y. If we had no antibody, we saw nice LNGFR, but no binding of the human antibody or to human IgG. But when we had rituximab, we saw a very nice labeling of the cells that correlated with LNGFR level. And so next, we went on to test this in human tumor xenograft experiments. So first, focusing on the left, this is an example showing this is a NOM6 cell line engineered to have HER2. And you can see if we just added the adapter, we get pretty rapid induction of tumor growth. But when we had the SNAP CAR T cells plus the adapter, we do see some anti-tumor activity. However, I do want to mention that while we see significant activity, it still wasn't clearing the mice. So we thought we had some work to do with this. At the same time, we also characterized the rituximab Fab, and similarly, we saw some activity from the Fab molecule with the CAR T cells compared to just adding the adapter alone. And from this, we went back, and one of the things we did was we looked at the CAR T cells in the animals. And I'm showing now, these are just our gating scheme for looking at the human T cells in these mice. And you can see that we were actually, with a full-length antibody, losing a lot of the CAR T cells in circulation. And this compared to if we added the Fab, we actually saw maintenance of that same CAR T level. So we're seeing a full-length antibody effect. And our thoughts kind of came down to, is there some Fc function from the antibodies that is actually leading to these cells getting killed off in the mice, whereas we don't see that with the Fab fragments. And so one of the things that led us to think that was, even in that first experiment that I showed you, where we had just one day later, we injected in the antibody and then the CAR T cells, and then saw this staining, this labeling or assembly of the CAR with the antibody. One thing we noticed was that while we're getting this great staining, we are actually, even after one day, losing about half of the CAR positive cells in the mice. So that led us to think that, yeah, there could be some Fc interaction happening. And so we came up with a plan to instead try to block the Fc receptors as a first pass using intravenous immunoglobulin. So the idea being to saturate receptors. And so we performed a very similar experiment where we did loading with the antibodies, and except we also injected in IVIG or didn't. And you can see that when we actually look at LNGFR in the mice, Now we have some loss when we don't have the IVIG, as before, about half the cells, even 1 day later. But with the IVIG, we do see retention of the LNGFR CAR positive cells in the mice. So this was very promising. We went on to scale this up and apply this to human tumor xenografts again. And again, using the NALM6 HER2 tumor cells, injecting in the CAR T cells along with IVIG, and then injecting in the Herceptin adapter every 3 days. And you can see again with adapter only or SNAP CAR T cells only, we see some pretty rapid tumor growth. But now when we add the SNAP CAR T cells plus the adapter with the IVIG, we're seeing nice reduction in tumor growth and elimination of tumor in the majority of animals. And this compared very well to a standard anti-HER2 CAR, usually the same binding domain. So this we're very excited by this activity. But that kind of led us to think about a couple different things. One, so we're using the adapters here to really target CAR T function, but we started to wonder, can we actually get additional function from having just the adapter alone? So universal CARs overall are a more complicated therapy. We have to generate the T cells plus the adapters. So ideally, we can really get even more activity from all the parts. And one way to do this could be to have more anti-tumor functions from the Fc domains of the adapters in addition to the CAR T activity. But at the same time, we also have some early signs that maybe we're having anti-CAR T functions from the Fc functions from the adapters. So we really wanted to dive into this more and kind of tease this apart and try to boost the anti-tumor functions and try to prevent the anti-CAR T functions if possible. And so we, still focusing on HER2, kind of came up with some different designs for adapters. And so one of them is, in addition to using Herceptin or trastuzumab, to actually use a different HER2 antibody that binds to a different epitope, pertuzumab. And so these would potentially have different Fc functions as well. And then we also wanted to look at, could we do some different methods of conjugation? So we were doing random lysine conjugation, but we were also interested in site-specific conjugation, I think, for these different Fc reasons, but also could we actually use this to get a more homogeneous drug product as well? So we had kind of a joint interest in that. And it was about this time that we were connected with SeromYx and learned really about their exciting platform for characterizing the Fc functions and Fc receptor binding profiling for different antibodies. And so we thought that collaborating with them would be really the best way to kind of get at that question. And so we had three major questions. One, what is the Fc binding profile of these different anti-HER2 adapters with the different conjugations? So with the random conjugation, with the site-specific conjugation versus the wild-type antibody. And we were looking at a couple of the FcR gamma receptors and also the neonatal receptor, which is important for recycling. So ideally, we could characterize these different ones with those modifications. And then we're also wondering, in addition to the Fc receptor binding, what are some of the different functions that these antibodies carry, both in interest for anti-tumor activity, but also then do these actually target the universal CAR T cells? And so some of the functions we were looking at were ADCC, ADCP, ADDCP, so macrophage activity, neutrophil phagocytosis, as well as on eosinophils, complement, and basophils as well. And so we started this program with SeromYx, and they did some FCR function profiling on the on the different adapters. And so I want to show you, excited to show you this data where we had our first looking at trastuzumab, we had the wild type antibody, then with the, in red, we have the random lysine conjugation, and then we have the site-specific conjugation in purple. So we see that the random conjugation is able to maintain that receptor binding, whereas the site-specific conjugation abrogated it. And overall, the neonatal receptor binding appears to be largely maintained with the random, but slightly affected with the site-specific. And this also held up when we look at pertuzumab, so very similar, except maybe less of a reduction in the neonatal binding here. So this, I think, to me was very exciting because we already had some variability in how these were acting. And so we could possibly use this different modification or this different conjugation strategy to change some of the activity of the adapters. And so in the next phase, we wanted to look at, so that's great about the Fc receptor binding, but how do these actually affect the functions? And the exciting thing about the SeromYx platform is that they can do this in the context of actual antigen binding. And so all of these different functional readouts here are in the context of the HER2 binding. And so it's a lot of data here, but I'll walk you through the highlights. So we saw that as expected from the Fc receptor binding profiles, we saw that the random conjugates really largely maintained the function of the wild type antibodies. And there is some variation between trastuzumab and pertuzumab, but overall, the wild type for each compares very similarly with the random conjugation. So that's with the phagocytosis, as well as ADCC, so the NK activity, neutrophil and eosinophil activity, as well as with basophils, and finally with complement. And but when we get to the site-specific conjugation, again, we see reduction in those activities largely, although not for all of them. So the main reductions are really with the phagocytosis and with NK activity, and some reduction in complement from the pertuzumab, and less so from the trastuzumab. So I think it's an exciting amount of variability here, but also that we do see these large changes from these different modifications. And so the next phase is really to look at how these are affecting CAR T cells, and can we actually protect them more? And so we have some very preliminary data here, and I just was excited to, because it's hot off the press, so I did want to show it, but the caveat that it is preliminary. So when we look 24 hours later in the mice, we see that the site-specific adapter does actually retain even more of the CAR positivity in the mice. And when we've done a preliminary experiment, and this is using a different CAR T cell actually in a different tag, but it is using the random lysine conjugation as well as versus a site-specific. We do see some improvement in anti-tumor activity when we use that site-specific adapter. So I think there's a lot of promise here, but and excited to see where the next phase of the work goes there. And so with that, I wanted to kind of summarize our conclusions so far with the project. So SnapCAR is really a highly modular universal CAR that will covalently associate with BG benzoguanine labeled antibody adapters. These cells show potent activity both in vitro, but also in a number of human tumor xenograft mouse models. In addition to what I showed today, we've also we have some nice data with targeting CD20 in mouse models as well. We found that some CAR T cells are lost with IgG1 based adapters in mice, and that they can be protected by IVIG. The antibody adapters, that have random lysine conjugation, we've found through SeromYx profiling that they retain FCR binding and largely the FC functions of the wild type antibodies, specifically ADCC, ADCP, and CDC. When we actually look at the site-specific conjugation, we find that the antibodies retain neonatal FC receptor function, but they largely lose their FCR gamma 2 and FCR gamma 3 binding, but they do retain some of the, they also lose some of the ADCP, ADCC, and ACDC activities with the adapters. But there is some difference between the pertuzumab and trastuzumab. So I think it's important to really, for any adapter, you can maybe make some generalization, but really deep profiling on each adapter is really important, I think, for determining its activities. And so the FCs are really key factors in these universal adapter designs. And kind of wanted to end with where we're thinking about this going next. And really, I think the focus is on getting the CAR T targeting, but also adding extra functions and minimizing the anti-CAR functions. So different strategies to do both of these. And with that, I just want to thank the lab and SeromYx and our collaborators, the Dieters Lab, and you for your attention. And I'll hand over for questions.
Okay, thank you so much to all of our presenters. Very insightful and exciting work coming out of SeromYx, as well as the University of Pittsburgh. So yes, we're going to open it up to the Q&A. I would encourage the audience to please add any questions that you have. I can kick it off. So Jason, I saw, sorry, I'm just looking back at my notes really quickly. You mentioned that the antibody adapters with site-specific conjugation, they retain the FcRn binding, but then they lose the other bindings that you mentioned. I'm wondering if you have any like hypotheses of like why it is that they lose certain ones but then retain others, if that makes sense.
Yes, and I think this comes down to the site where we're attaching the benzoguanine motif. So I think that is key, and I think even now looking at structural analysis, this probably would make sense that we would lose that activity, so yeah, so I think it's all about the location of that modification.
Understood. So
yeah,
we
were confer with Jason, the FcRn binding site, Fc gamma receptor binding sites are non-overlapping on the Fc.
That makes a lot of sense. BRT Matal says, thank you so much for a nice talk. And then Anaya Mishra says that they are a microbiology student and want to know, how do Fc modifications in linker design influence and balance between CAR T cell specificity and off-target immune activation in such conjugated systems?
Sorry, can you repeat the first part of that again? So
Sure.
Yeah.
How do Fc modifications and linker design influence and balance between CAR T cell specificity? Does that help?
Yes, great.
Okay.
Yeah, so I think with the adapter design, having the different FCs, I think really it's a balance again between having the FC and then having the neonatal receptors recycle the antibody to increase the half-life of the antibody. So that will really keep it longer in circulation, which for an adapter, we like that because it means fewer doses. These are given by infusions, so we really ideally want to minimize the number of doses. But at the same time, could lead to having the anti-CAR T cell effects. So if the FC if these effector cells are targeting the CAR T cells and eliminating them, then that could actually be problematic to the CAR T cells. But at the same time, we do want to maintain the FC functions of the antibodies against the tumor. Maintaining those functions will be beneficial. And in terms of toxicity, I think you can think about the circulation time could lead to more, I guess, binding on cells that have lower antigen expression. So you could potentially get some enhanced toxicity by having that longer lived effect, or by having some, I mean, there are many known toxicities for Herceptin in general, as well as pertuzumab. So you could be getting some of those toxicities from Fc-mediated functions from those as well. And lastly, I know you mentioned the linker length. So that is kind of binding between the CAR and then the antigen, or the adapter. So that helps to determine the synapse distance between the CAR T cell and the target cell. And it's been shown that if that length is too long, then you actually don't get efficient CAR signaling. And so that is something, one area where we play around with the linker length, is to figure out how to optimize the CAR signaling.
Thank you so much. And thank you, Anaya, for your question. Gonna put it out there for anyone else who has questions, and then in the meantime, I will ask another. So I know you shared a little bit about like your future plans for this work, and I'm wondering, like, for the audience, like, how do you think that they can apply the work that you all have done, like, in their workplaces, regardless if it's like in an academic institution or like some of the people probably from CROs or pharma, like, yeah, how do you think that this would support their work? And I guess that's a general question, so if either of you have answers.
Yeah, I was thinking, Shashi, maybe this one is mainly for you, but I can also give a CAR T specific answer, but I can let you take it first.
Yeah, I mean, not being guilty of repetition, but what I think is safety and efficacy are both important. And what Jason's doing, what their approach is to de-risk both, right? And you don't want to lose a good partner at the same time. You don't want to create a whole storm and have issues to deal with, especially when you're going into NHP talks. I mean, this is even before first in human. NHPs as a waiting line, they're expensive, and they're necessary for IND enabling. And so you must have a approach where you're de-risking that development. And finally, it's about treating the patient. Finally, it's about getting rid of the cancer. And that's where you have to do your best to wipe out the cancer cell without collateral damage. So I know it's a very high level answer, and we know that these people have all kinds of funnels where they go through to de-risk safety and efficacy, potency assays included, the pan lab receptors included. But what people have not done yet with antibody therapeutics that have an FC is de-risk the FC, or to really partner with the FC. And that's where this is coming from. So yes, any developer, amongst the 90% of developers who have an FC in their antibody drug, please make sure you know what the FC is doing.
Yeah, and I also extend that to anybody developing universal CAR T cells. Please investigate the adapter and for all those extra FC functions and FC receptor binding. It's definitely a huge part of our current development.
Thank you so much for that. That definitely makes a lot of sense. I think that we're right at time now. So if there are any other burning questions, feel free to reach out directly to the Antibody email address. We have info@antibodysociety.org. Oh, just got one more. Do you want me to go ahead and say that? Sure. Okay. So there is a microbiology student named Crescioli who wants to know, what is the metabolic trade-off when increasing the half-life of an antibody that has significant off-tumor on-target binding? And you all can also see the questions in the chat if you want to read it for yourself.
Yeah.
Well, you wouldn't really want to do that. So if he has off-tumor on-target binding, maybe you can first try to address the issue and maybe optimize your antibody to improve the on-tumor on-target, and so reduce toxicity. Because of course, if you increase the half-life of these antibodies, you might, and you know that you have significant tumor on target, you will have toxicity. So you will first need to optimize your antibody to reduce that, and then you can think about extending the half-life. I think you need to, not always an extended half-life is the right choice. It depends on the context.
Yeah, I think that's an excellent point that Silvia makes. Also, just want to pick on the word metabolic. I don't think most of the burden is metabolic. I think the trade-off is mostly immunologic or disease progression. And you want a long half-life for several reasons: cost of goods, patient compliance. You don't want to have repeat doses. So, and you want enough long-term target engagement on tumor, on target. But if there's going to be some off target, then maybe specificity or dialing certain affinities or working with a different epitope for that, may influence some of these outcomes from keeping the same or a longer half-life.
Thank you so much, Crescioli. I hope that answers and helps your burning question. And last one, we have another one from Anaya. How are next generation engineered antibodies being optimized to overcome the immunosuppressive tumor microenvironment, especially issues like hypoxia and stromal barriers?
I can take a stab at that, and then I'll pass it to Silvia and then Jason. I highlighted very quickly that we have a paper that we published with Silvia Crescioli's lab, and she's one of the few investigators in the world who are not trying to hide from the FC. She's actually supercharging the FC by engineering. And the point there is you can have an immunosuppressive environment that gets completely configured if you have FC effector function that you utilize in a smart way. And we did see that in the paper, even for the IgE modality, Osborne et al., Nature Communications, it shows that we took an immunosuppressive environment and utilized FC engineering, which drew from macrophages that were anti-tumor, didn't skew the axis away from the pro-tumor macrophages, and made it an excellent efficacious drug-like molecule, right? So Silvia, go for it.
Yeah, I totally agree with you. And also another methodology could be to find other ways to recruit the right type of immune cells to transform a cold tumor into a hot tumor. So now there's lately there's been a very nascent trend of antibody conjugates with payloads that actually stimulate innate immune cells. These are called ISACs, immunostimulated antibody conjugates. So that's one of the next generation's modality. It's still early stage, so we don't know how we will go, but that's in principle is a very clever method.
And I think those are great responses. I don't have anything to add there.
Great. Well, thank you all so much. Thank you, everyone who attended and asked really insightful questions. Really appreciate the SeromYx team, University of Pittsburgh, Shashi, Silvia, and Jason for presenting today. So we'll have an on-demand version of this scientific briefing available very soon. We'll share a link to everyone who registered via email, and we'll also upload this to our YouTube channel so you can check it out on demand. And feel free to watch this or any of our other on-demand webinars whenever it's convenient for you. Thank you so much, everyone, and have a great rest of your day.
Thanks.
Thank you.